Welcome to the Research Cyber Infrastructure Symposium. Um, in this fourth installment of our talks, we have Antoine Regimbeau, who received an engineering degree from École Polytechnique in Paris, where he majored in autonomous systems and signal processing. After working as a software engineer for just a couple years, he began a PhD at Nantes Université in 2019. Um, he earned a PhD in computer science in 2022, focusing on modeling microorganisms in the ocean and using omics data. In 2023, he was awarded a Simons Postdoctoral Fellowship and joined the DeLong Lab as a postdoctoral researcher since April 2024. Welcome.
Thank you. So today I'm going to talk to you about how we go from DNA to microbial ecology results and how we model community. So as Ron said, I'm a postdoc and I'm studying microorganisms in the ocean. And one of the reasons why I keep studying those microorganisms is because of this book from Falkowski, which talk about how microbes made the Earth habitable, or they terraformed the entire biosphere so that we have life as we know it now. It's a very interesting book. I highly recommend it. And another reason why, few other reasons why I'm studying those organisms in the ocean is because they are actually quite important. So they made more than 2/3 of the oceanic biomass. They are responsible for more than 50% of the oxygen present in the atmosphere. Of course, they are at the base of the food chain, and they are a huge carbon sink. So to study those microorganisms, we are going to look at their DNA. So you might be familiar with what it is, but let me just briefly present it. So strands of DNA are composed of letters A, T, G, and C. We call them pair, and we measure the size of DNA by the number of base pairs it's composed of. On the DNA strand, you have coding sequence, which we call genes, and non-coding one. We are more interested, of course, in coding sequence. And everybody knows that DNA shapes into chromosomes. You must be familiar with the X shape of chromosome, but when we are studying bacteria, we are encountering a circular chromosome like you have on the right. Very briefly, so the DNA was discovered in 1969 by Friedrich Schmitter, but at that time it was not believed to be the support of genetic information. We have to wait 1944 for an experiment to prove that, and afterward 1970 for being able to read DNA. So just a bit of language here. We sequence the DNA, and by sequencing DNA, we have reads, and reads are basically chunks of DNA. And today's technology allows us to get reads from 50 base pair to 100 kilobase pair, even more if you have the right tools. So that's quite huge. So what we have at the DeLong Lab is a reads, so sequence of 150 base pairs, and in a particular time series that I will present later, we have 10 million reads per samples. So what you have on the right is 2 examples of reads. And if I gave you read like this out of the blue, frankly, it doesn't make sense. And that's a statement that Noam Chomsky made when he was talking about linguistic theory, but I think it still applies to bioinformatics. Maybe it's because DNA in itself is also a language, but it's true that if we only consider raw reads, it doesn't make sense at all. And in my opinion, we need to process the data into product and then process it again into sense. And there is also one hypothesis that we often forget, is the fact that this data that we have, we are hypothesizing that it's characterizing a system. And for us, the DNA we are gathering characterize the community of microorganisms that are in the ocean. So now let's talk about how we actually get that DNA. So I'm going to talk about the Hawaii Ocean Time Series, or HOTS in short, for short. And it's an old time series that's been going on for quite some time, happening up north of Oahu. The time where we have actual DNA data is shorter. It's only 8.5 years, but that's already pretty good for DNA time series. And the way we do it is that we go to the station up north of Oahu called the Aloha Station, and we basically toss a CTD sampler. So CTD sampler is a bunch of electronic tools that measure conductivity, temperature, and depth. And to those tools, we attach a bottle of water, the black stuff that you can see on the picture here. And we open the water, we fill the bottle with water at the different depths. And with that water, afterwards, we filter it and we sequence it to get the DNA that were in the water. We have two size fraction, the very small one called the viral fraction, and a little bit bigger one called the cellular fraction. So it's still small, right? It's the order of magnitude of the micrometer. So in this time series, we have sequence of 150 base pair. The hypothesis is that it's a fragment of genomes of the organism present there. And we have more than 1,400 samples, which totalize 16 terabytes of data. However, as I told you before, those data doesn't make sense. So we are going to process it into a product, and our product is called contigs, which are basically just longer sequence of base pair, again, that are fragment of genomes. And so the process of doing that is called assemble, and in bioinformatics, assembling is the cornerstone of the field. And if I can do an analogy here, just to give you a grasp of the problem we are dealing with. So 150 characters is roughly 3 lines of a book. So imagine I give you a bunch of 3 lines and I tell you, well, now try to assemble that in the Lord of the Rings trilogy, which is roughly 3 million characters, where a bacteria genome is 1 to 10 million bases. So that would be the problem, the issue you are dealing with. However, as we are sequencing not one organisms, but the entire water mass, we don't have only one genomes. We have hundreds of them. So that would be really trying to assemble 3 lines into libraries with hundreds of books. So let's have a look at the naive way of doing it. I give you here a data set, few sequence of character, and a naive approach to assemble that would be to look for the longest common substring in each pair, then assemble those pairs, and then repeat the process. So quickly here, you have the common subscreen, the size of the common subscreen, then you assemble the green and the red pair, and you repeat the process. At the end of the day, you have your sentence. However, the complexity of this algorithm, because of the for each pair, is in quadratic in number of sequence you have. So for us, it's quadratic in number of reads. And remember, we have 10 million of reads per sample. So that's not a really good algorithm. Fortunately for us, there is graph theory. And this is a De Bruijn graph that we are using heavily during the assembling process. They are around since 1894, thanks to Camille Sainte-Marie, and it's been formalized in 1946 by De Bruijn. It's used widely in information theory and interpretation algorithm, but since the 2000s, it's also used in biology or bioinformatics. It was introduced by Pesner in this article, and like the first line of the article was comparing biologists to children, saying that the way they assemble genomes looks like children assembling puzzles. The only difference would be that the genome is way larger than a puzzle. So he proposed to biologists kindly to use the De Bruijn graph. So what's the De Bruijn graph? It's a very, I think it's a very elegant data structure that allows us to solve this problem very efficiently. And the way to do so is that you're going to need to build the graph. And in this graph, the nodes are all the subscreen of a given size. And then you're going to build each edge between the graph between nodes that share a prefix or a suffix of size k minus 1. So if we go over this example again, we list all the substring of size 4, then made that into nodes, remove, of course, the duplicates, and then add the edges. Now, to get your sentence, you just have to go through the graph. So I think it was pretty easy, even if you compare to the previous methods. Now, if we look at the complexity here, well, the overall complexity of the algorithm is linear in number of reads, which is a big improvement. And the reason why is because creating the nodes is linear in number of reads, because you just have to go over your entire data set to extract the substring. And then creating the edge, you can be fancy and use a hash table to linearize the complexity here. So that's a big, big improvement. So that allows us to, from the 3 lines, transform the 3 lines into libraries. However, it's not that simple. And while in a book, you have more than 30 characters, 30 different characters, when we are dealing with DNA, you only have 4 characters, A, T, G, and C. That means that you will have a lot of repetition in your reads, which will give you a way more complex Kaplan graph. So all in all, we won't be able to build the library, build all the genomes we have in our data, but rather we will be able to build, to reconstruct only pages of book, which are the contents, right? It's just longer sequence. Not the entire book, but just the pages. So back to that time series in number. So the time series is at the beginning 60 terabytes of data. That doesn't make sense. And we process it into less than 1 terabyte of data that this time does make sense. And to do so, we are using heavily the HPC cluster core. And I'll give you just a brief example of how much we are using it. So that's the entire workflow for the hot time series. So we go from read to contig, that's what I present to you, and we are using COA here. And then we go from contig to genes on that branch. This is just detection of genes, so we don't need COA, but afterwards, there is this step when you want to go from genes to abundance table. And to do so, you are taking every reads and you are mapping them to genes. So that's a very computationally intensive step. And this time we need COA again. Or you can go the other way from contigs to mags. So if we take back the analogy of books and genomes, going from contigs to mags would be going from pages of books to books, right? Mags is a fancy way to say genome that are assembles from contigs and not from contigs from various genomes. So there might be error in them. And then you go from mags to abundant table, and to do so again, it's mapping the read onto mags, very heavy, very computationally intensive process. And I made the computation thanks to John Edley that is doing all this stuff. And processing the entire time series is roughly 1.4 total CPU hours delivered by COA for the year 24 to 25. And I think it's huge because I had no idea we were using that much CPU hours to do so. And the entire process is taking more than 60 CPU years as I transform the hours into years, because at some point there's no point of talking about hours. It's so big, it doesn't mean anything. So that means if you had a personal computer, it would take you like a year to process the entire time series. So we highly rely on infrastructure such as COA to do our work. That's one aspect of the process. The other aspect is also the fact that we are drastically reducing the size of the data we are dealing with. We go from 19 terabytes to all the way down to 18 gigabytes, so that's a factor of 1,000. And at the end of the day, we have a big table of size 53 gigabyte, where you have the genes, but also the annotation. Annotation of the genes are either their function, because we are able to recognize the genes, or where they are supposed to come from, which organism they are supposed to come from. So that's all combined in one big table. Now, what can we do? So the first thing I did with the hot time series is try to apply a dimension reduction algorithm on it. So I had more than 1,400 samples in more than 60 million, 16 million dimension. That is how I need 60 million genes to describe the entire data set. And that's the result I pulled is that you can see a cluster of different depths, like the lower depth, but you can also see the cluster coming from the virus, because remember, we also have the viral fraction and the 5-meter viral fraction. And the big cluster in the middle, if you just take that and apply the reduction algorithm on it, you can see that there is a nice continuity in the space on which it's projected from the 5-meter to the 250 meters. And I think that's nice because it's showing that the genes continuously, the gene contents of our samples continuously adapt to the depth. On depth, there is also one nice result we have, which is the structure of the genes. So if you remember, the genes are composed of A, T, G, and C. And if you can focus on the GC content of the genes we have in our sample, you can see plots like this. And you can see two shifts, one first around 100 meters and the other one at 4,000 meters. And why one is explained because of the availability of nitrogen in the water column that is low at the surface but higher below, hence the GC content that is rising. The other one is still an open question because we just saw it. So we have no idea. Some of us have ideas about that. Whereas it might be because of the depth, pressure, and then the adaptability of the organisms at high depth. But it's still an open question. So that was for the broad view of the data sets. Now, if we want to study the community that is there, I'm going to show you a few results that rely on what is called the Bray-Curtis dissimilarity. So if you're not familiar with this dissimilarity, it's a way to compare two samples. So let's say we have sample A and B. The dissimilarity will be 1 if you have no sample of A in B and no sample of B, no member of A in B and no members of B in A, and it will be 0 if both samples have the same members at the same proportion. With that, we will be able to study season and dynamic. So the season will be just the average over time of the dissimilarity for a given lag. And the dynamic would be just the dissimilarity between two time consecutive samples. So that's the results for the seasonality of the community. On the left, you have the dissimilarity computed on organisms. So the member of the sample will be this time organism. And on the right, you have the same thing, but this time based on genes. And you might think that if you have the results with the organisms, you will have the results with the genes. Well, it was not that obvious because microorganisms tend to have genomes that are highly variable. So we were glad to also find this seasonality with the community if you consider genes instead of organisms. And for the dynamic of the community, so this time it's only based on genes. And here I plot for depth so that you can see how it looks like. But I want to draw your attention on the fact that there is, among those 4 days, there is a slight tendency of the dissimilarity to increase over time. And if I do a regression linear on all the depths, I have that kind of plot. So there is the tendency to higher dissimilarity over time is present in all depths. And I think it's important. It means that the more you go forward in depth, the more energy the community uses to change, to adapt to its environment. So it raises the question, if the community is changing that much, what about the function of the community? So if we look at that from an ecology perspective, it will mean what are the service of the community to the ecosystem, to the biosphere. And so I did the same thing, but this time, instead of looking at genes, I was looking at function. And I was glad to see that this tendency is not present when you look at the function, which means that the function of the community is rather stable. Its members are not, but I mean, its genes are not, but the function, the set of function at least, is quite stable. So it raises two questions in which I'm particularly interested in, which are, the first one is studying the driver. So what pushes the community to change its gene while keeping a relatively stable set of function. And the other one is trying to study what I would call the tipping points. If how much can a community change in terms of genes without changing its function? And I think those two questions are pretty interesting and pretty important given the whole the whole climate change climate we are in. Yeah, so that was it for the whole study of the community and the Hawaii time series. Now I want to talk a little bit to you about models. First, I will introduce two concepts. The first one is emergent property. That is the property of a system that are neither predictable from nor reducible to its individual components. So let's take water, for instance. It's composed of H2O. However, from that, you won't be able to say that water is transparent. The transparency of the water is an emergent property of the water itself, not of its components. And also the abstraction of the system or the levels of organization, which is basically the preciseness at which you want to describe the process that are in your system. If you have high computational power, you might want to very precisely describe the system, but how otherwise, depending on the question, you might take a more global look at your system. For instance, let's take a very simple example. You want to know how long it takes to drive from here to North Shore. Well, if you know a lot about car, you might be tempted to look at each subsystem of a car and say, I can model each subsystem and then make them communicate. And with that, you will have the results. But there is also another way to model that and to answer the question. It would be, well, you have a car, you don't care how it looks, but you have the emergent property, which is the mean velocity and the gas consumption. And just with that, you're able to say, well, if you want to go from here to the North Shore, it's going to take you 30 minutes and less than a third of a gallon. And that's done. So from one model, very simple, you're able to answer the scientific question. And so when you go back to our community of microorganisms, there is a lot of ways to model it. One way would be for each organisms, get a model, right? So you have 100 of organisms, that means you will have 100 of models. However, to me, that's not the right way to do it. And the reason why is because first, it's going to be impossible to compute and to simulate because it's going to require a lot of power. And also, to parameterize those models, you will need a lot of data that we don't have right now. Like how microorganisms are interacting is something very complicated to infer. So to me, that's not the right way to do. So now we'll present how I would do it. What I will do is basically just take the whole community and say, well, I don't want to know how every individual are interacting. I just want to know how the community is behaving. So let's say that the community is one big organism, and let's study this big organism. This is called the supraorganism approach, and it's used also in microbiology. And that's what I'm going to do. So first, study the system and look for its emergent property. And in my project, I'm doing that, but this time not with the hot time series, but with another one. If you remember, the hot time series, we're going at sea and sampling 1 point in the ocean every month or so. And here it's a different time series where we follow the water mass. I mean, not me, but the crew of the cruise follow the water mass and sample the water mass every 3 hours. The idea here was to observe the same community over time in the water mass, which is something quite complicated, and they did a fantastic job. And so the crew did 2 legs from with the 10 days apart, and they tried to. I mean, the hypothesis is that between leg 1 and leg 2, we are still following the same community. So that's the hypothesis of the cruise. We are observing the same community in leg 1 and leg 2, and we get its data. That's the data I have. So remember, this is again not the data. This is a product. John had to assemble that from reads, and I have roughly 100K genes and 30 time points per leg. Right, it's 2 independent legs. And what I did after that is try to look for co-expressed genes. So I did a correlation graph based on Spearman correlation. And then I run a clustering algorithm. It's called InfoMap. And with that, I have different cluster for the leg. I did that with the 2 leg independently, right? There is no, I didn't put information from leg 1 into leg 2. And the result I got was that I had 5 clusters in each leg. And here I plot only 3, and those to me are the most interesting. And the reason why is because if you look at leg 1, to focus on leg 1, you can see that the cluster blue, for instance, is picking its expression during nighttime, while the green is picking early in the morning, and the orange is picking in the middle of the day. That means that we are really capturing 3 different processes of our community. And it's the same on the leg 2, right? We have those same type of behavior. So with that results, I wanted to investigate more and look at the composition of the cluster, if they match. And the answer is yes. So I looked at the Jacarandex of the cluster, the cluster from the 2 leg based on genes. And what you can see here, for instance, is that the cluster 1 from leg 1 corresponds to the cluster 1 of leg 2. And it goes the same for the 3 clusters that I showed you on the previous slide. So that's what we are showing. And in a sense, it validates the fact that we are observing the same community between the 2 legs, and probably that the 3 groups we are observing are characteristic of our system. So back to our community. From that, what I infer is that the emergent property of my system would be that there is 3 groups of co-expressed genes. Now I need to transform that to formalize that into model. And what I used is called genome scale model. And the idea here is you have the gene, you have the DNA, you know that this gene is encoding for enzyme, and you know that this enzyme is catalyzing a reaction. And that's the whole process here. It's reconstructing what we call here a metabolic network. So you go from a set of genes to a graph that represents the metabolic ability of your set of genes. And as this, the reason why we are using graph or network is because on some other assumption, we are able to transform that into linear equation. And this in computer science is a goal because it's super easy to simulate if you don't have too much, but we don't have too much. And because you have very cool solver that can do that for you. So the typical size of a model of such a model for a bacteria would be 2K reactions. And when I look at the entire community, I'm happy because all the model together, they are totaling less than 10K reaction. I'm at that point right now, and I don't present more the genome scale because basically I present that very quickly, but the way you build the genome scale model from your set of genes, there are a lot of uncertainties, and right now I'm trying to assess these uncertainties, trying to know if I'm actually modeling the set of genes or if it's some other artifacts. That's where I'm at now. So I guess the take-home message is for this presentation would be that we definitely need computational power and infrastructure like COA to process biological data. Otherwise, we will be like child playing with puzzles. The community of microorganisms that we are studying at Station Aloha might be changing more and more in terms of genes. However, the function of the community does not change yet, and I think that's a good point, and we should work in order for it not to change its function. And last but not least, that when you want to answer a scientific question, you need to spend time of choosing the correct abstraction, because with that, you will be able to leverage various limitations like computational power or even the need of data that you can have. I thank you for your attention, and I'm happy to take questions now.
Anyone online, if you have any questions, you're more than welcome to unmute yourself or just drop your question in the chat.
Have you looked at AI models comparing microorganisms to certain categories, like using attention mechanisms instead of just experiments correlation?
Again, please. I didn't hear.
Some of the AI models that let you assess attention between different components, let's say a microorganism and its correlation. Could you do that? Have you looked into using AI models rather than just experiments correlation or other statistical methods?
I mean, if we take this step, you want to instead of going from the data to the correlation graph, apply any machine learning algorithm to get that.
Right. Both these graphs, correlation and clustering.
Yeah, so I didn't do, and I'm not very fond of AI technique because you don't know why they will gather the stuff together, and I need this link in order to build the process. So I tried different things instead of just a correlation graph, Spearman correlation. For instance, I look at the mutual information between genes. That might more look like AI stuff. The issue here is that the data we have is too noisy, and the mutual information will drop drastically every time you have like 3 time points that will not be together. So it was not working. With that, using mutual information, I would have the result that the 2 legs were not looking at the same community. So I fall back onto something that it was a bit more robust. And also one thing about AI is that, to me, at least, it's reproducing something you already have, right? It will look something that it already knows in your data. Whereas here, well, I don't know anything about how it's working. That's why I'm using such technique. The clustering algorithm is also, it's not AI per se, but it's also machine learning algorithm when you try to compress your whole data sets. But there is AI technique to build model after that, like genome scale model. But again, because of the uncertainties, you will end up with a model, but you will have no way to prove that this model is actually representing your community. That's why I'm not using that much.
Yeah, I get that. You have to experiment with the model, like apply to downstream tasks. That's the only validation. Again, Thanks.
So you see the genes changing. Is there any way to kind of see where it's expected to go next? Over time,
Yeah.
hypothesis. So where is it going?
I mean the stationary web stuff?
Yeah, we're gonna go to like unlimited complexity or what? You know?
It's not so much complexity. It's to me, I interpret that in terms of energy. So if you think about the community as a system that is changing under environmental pressure, the fact that between two time points it's changing more and more, that means it has to spend more energy into adapting, right? At some point it won't be able to adapt. And I think that's my opinion on that, is the fact that at some point it won't be able to adapt. And what we would see is that the function of the community will change. So it won't go up until
Like the diversity is decreasing, and then eventually it'll collapse into some steady state, or
not steady state, but to some other intermediate state, and it's going to be fluctuating, and not only the genes but also the function. Yeah, that's what I think it will happen.
so also like, is that happening everywhere?
No.
Is that just a spot in the deep ocean, or is it just the one spot?
Oh no, it's just that what we have at Aloha, but it's hard to say it's happening everywhere because we have that type of time series only there. I mean, I could apply that to other time series, but I don't have my hand on the data.
I guess if you have a model, right, you can get data, try to fit it to that model. If you think it's
yes,
variable, if you can keep variables.
Yeah, that's true. That's true. I already did some same type of research from another data set when I was a PhD student, and what we observed is that it was the stability of the system were not the same everywhere on the planet. So maybe we are indeed sampling at some point where it's changing a lot, but across the whole planet, for instance, it was way more stable up north and south, like in cold water, for instance.
The gene was more stable or the function was more stable?
No, this time we were talking about the function. Yeah. As long as we are, I mean, in my domain, in my work, as long as I'm talking about model, I'm talking about function, because I'm not able to model the actual genes. There are way too many genes and it's way too complex.
Any other questions from anyone online?
Nothing online.
Okay. Thank you so much for your time. This was a really great talk. Very nice to hear, what our researchers are doing with COA. So thank you so much for sharing. I appreciate your time.